Okay, thank you everyone, and I hope that online you can also hear me. And it is my honor this afternoon to introduce our keynote speaker, Professor Gwen Robins-Shug, who has kindly traveled here all the way from the United States to talk today to us. Presenting on paleopathology, lessons from the past about climate change and human health. Sorry, I need to admit her into the chat in 1 second. As you can see, Professor Gwen Robins-Shug has been a full professor at the University of North Carolina at Greensboro since 2022, I hope I'm correct. And before this appointment, she rose through the ranks at Appalachian State University, where she spent approximately 16 years, becoming full professor in 2017. And her research bridges biological anthropology, archaeology, and paleoclimatology to examine how past societies experienced and adapted to climate change, often in ways that echo contemporary global challenges. Her career has meandered through several interesting spatiotemporal contexts. In particular, she has conducted extensive work in South Asia, using human skeletal remains to reconstruct health, stress, migration, and resilience during periods of ecological and social upheaval. Her research illustrates the axis of climate change with the human past, present, and future, and her work has been globally recognized, particularly through publications such as two of her books, Bioarchaeology and Climate Change, A View from South Asian Prehistory in 2011, and another, The Root Leg Handbook of Bioarchaeology of Climate and Environmental Change in 2020, just to name a few. And Professor Robins-Shug is widely recognized as the leading scholar shaping the theoretical foundations of the bioarchaeology of climate change, pioneering an integrative approach that foregrounds the human experience, inequality, and resilience in the face of environmental disruption. So please join me in welcoming Professor Gwen Robins-Shug, and we're very grateful to have you here today as the kind of pillar of the bioarchaeology of climate change.
Thank you so much for that kind introduction. I'd like to thank Valentina and Julia for inviting me to be here today. I actually lead a study abroad program, and we begin in Italy a week from now, so it's nice to have some time to relax a little bit before that happens here in Italy, which is such an amazing country. So I want to talk with you today about a theoretical framework that Jane Bijkstra and I have been trying to promote across bioarchaeology and paleopathology, which is the One Paleopathology Framework, which is basically an interdisciplinary framework. Excuse me. Oh. It doesn't want to, there we go, okay. An interdisciplinary framework for using deep time evidence to reconstruct complex interactions between humans, animals, and their environments. And ultimately, we would like to inform contemporary health and conservation strategies. And so our goal is to think about how today Earth's mean surface temperature has already increased by a single degree Celsius, and that's over the past 150 years, but mainly in the last 4 decades. And we are actually scheduled to perhaps even reach 1.5 degrees Celsius this summer. And we know that over the next 80 years, without an immediate and dramatic change in behavior, Earth's mean annual surface temperature can increase anywhere between 3 to 8 degrees Celsius, levels that have not been seen since the mid-Miocene to the Eocene. And the risk of this kind of global warming for human society, it's a very complex situation with interacting hazards that are compounded and potentially amplified by human responses. So one difficulty with actually adapting to anthropogenic climate change is uncertainty about how it will affect us. And we use the past to build prediction scenarios. So you can see here how the temperature that we're expected to experience by 2100 is actually taking us back to a time in the mid-Miocene when our order went through dramatic changes, and the Eocene when primates first evolved. So weird. Sorry. A little strange. And there are myriad ways in which climate change is predicted to impact human health, from temperature-related impacts to extreme weather events, nutritional insufficiency, and changes in vector-borne and respiratory infectious diseases. Extreme heat and unpredictable weather and ecosystem change will impact human health, and climate change is an indisputable threat, but it does not exist in a vacuum, because human societies are already confronted with rising social inequality, political and economic uncertainty, particularly in my country, and a cascade of concurrent environmental challenges, including the sixth mass extinction, emerging and re-emerging diseases, and antibiotic-resistant pathogens. And additionally, of course, all of these forces operate on human health within a strongly influential historical and sociocultural context. For example, with COVID-19, the pandemic starkly illustrated that humans are a biocultural organism. While understanding pathogen biology, transmission routes, and virulence is crucial, these biological aspects alone cannot adequately prepare us for an era of emerging and re-emerging infections. Recent experience has shown us that the same pathogen yields vastly different health outcomes due to a complex interplay of sociocultural and historical forces. And the recognition of the crucial role of social sciences in epidemiological modeling and pandemics research is gaining prominence on a global scale. As Francis Collins noted in his exit interview in 2021, when he retired as the director of the National Institutes of Health in the United States, he expressed his astonishment at the escalation of culture wars to the point where many Americans struggle to distinguish fact from fiction, which of course is only getting worse now. He emphasized that social science is essential for ensuring a more stable future, and one of his biggest regrets as the director was not allocating more funding for social science research during his tenure. So with COVID in the United States, for example, stigmatization, spite, structural inequality, misogyny, and racism were really important aspects of our response to the pandemic and the health outcomes. The virus was deadly, but the human response was deadlier. Sadly, our current government is cutting vaccination programs, funding for scientific research, and initiatives to fight global inequality. So what should happen for humans to navigate these complex challenges is that we must model societal adaptive capacity and social sustainability. And archaeology is particularly important in this understanding, because one way to think about and make predictions about the future is to understand how humans navigated these types of complex challenges in the past. So one paleopathology then combines observations of past climate change with insights on human and environmental health from archaeological remains to address these questions about how to create an equitable, sustainable future. We require high-resolution paleoclimate data to infer relationships among environmental change and human health over the long history of the Holocene interglacial period beginning 12,000 years ago. Preceding the historic era of written records, there have been three major rapid climate change events that have been documented at 6,200, 4,200, and 1100 BC. We've also experienced extreme climate change events historically. Of course, you all are here studying the late antique Little Ice Age, and we had the medieval warm period as well. Most of these events brought cooling and increasing aridity, which is not in direction akin to a modern global warming challenge, but these events are often similar in the magnitude, intensity, and rapidity of the change, if not the direction. Environmental and climate changes have been principal drivers for the coevolution of humans, animals, and our pathogens. Infectious pathogens benefit from large-scale landscape modifications associated with agriculture and pastoralism, which began around 9,000 years ago. This is a paper that I published with some colleagues of mine in 2019 that demonstrates landscape transformations across the globe over the last 9,000 years. Agriculture and associated lifestyle changes, settled farming communities, close contact with other animals, high population density, less varied diet, these all created significant challenges for human communities and significant opportunities for pathogen evolution. Worldwide, human skeletal remains from the first farmers have demonstrated evidence of the relationship between anthropogenic ecological changes and changing disease ecology, including the emergence, evolution, and global spread of diseases like malaria, tuberculosis, leprosy, brucellosis, leishmaniasis, salmonella, smallpox, hepatitis B, treponemal infection, and Chagas's disease, among many others. Ancient human-environmental interactions shaped malaria, for example. This disease is a global health concern, which has often been assumed to be linked to farming practices in the past, which altered insect vector habitat. Plasmodium falciparum was a leading cause of morbidity and mortality in ancient Rome, for example. But a paleopathological analysis of settled hunting and foraging populations in Southeast Asia has revealed that agricultural expansion is not the sole driver of vector-borne disease. Malaria posed a significant health challenge in tropical northern Vietnam 9,000 years ago, according to genetic evidence for thalassemia in human skeletons. And this is close to the initial occupation of human bodies by this pathogen. This population was not clearing land for farming, but rather population growth and reduced mobility fueled the rise in morbidity. Today, epidemic malaria is a growing concern as the pathogen spills over from mosquitoes that preferentially bite other primate species, but which are now causing disease in human communities. Urbanism has contributed to infectious disease ecology for millennia, though. High-density human populations, poor sanitation, crowded living conditions, and widening socioeconomic inequality facilitate the spread of communicable pathogens from person to person, compounding the risk for endemic and epidemic disease due to contaminated air, water, and soil. Domestication, deforestation, and environmental degradation increase opportunities for zoonotic spillover, and regional trade networks can provide new reservoirs for human pathogens. Bioarchaeology has demonstrated that some emerging and re-emerging medieval epidemic diseases had their roots much earlier in human history, with the first interconnected cities of the Bronze Age in South and West Asia. Around 4200 BC, there was a rapid climate change event that coincided with massive population reorganization in the Indus civilization, for example, in South Asia. People from all over South and West Asia migrated to urban centers initially during this climate change event, and many of these cities held 20 to 40,000 people and were built on sterile soil, laid out in a grid. As their trade networks expanded, new diseases may have entered the subcontinent. Mycobacterial pathogens leprosy and tuberculosis are seen in South Asia for the first time during the Indus civilization. And we know that this disease spillover event directly preceded widespread site abandonment at some of the cities of the Indus civilization. After 200 years of increasingly arid conditions and shifts in the balance of power in their trade relations, Indus people left these cities and moved out to small villages. They returned to mixed economic systems that included subsistence farming, hunting, herding, and gathering wild food. Skeletal evidence for leprosy occurs in 9 individuals buried at the city of Harappa and 1 individual in a far-flung rural outpost at Balatal. But most of the skeletons from this civilization have yet to be examined for pathology. Something very important to emphasize is that Leprosy, tuberculosis, and interpersonal violence were most common in the Indus civilization sites in folks who were buried not in the city cemeteries, but in marginal burial areas located near sewage drains, for example. In this deposit outside of the city of Harappa, it's a marginal burial area. It's located near a sewage drain that exits the southern wall of the city in a very atypical deposit of skulls that are piled and a single decapitated prone skeleton, which were all lying underneath another deposit of piled ceramic vessels. Thus, it appears that the risk of leprosy and interpersonal violence was structured by social divisions and inequality, not migration or conflict over resources, as popular authors who write about climate change today would have us believe. There are health benefits associated with living in smaller communities and maintaining adaptive diversity and flexible subsistence practices in the face of a changing environment. This is evident among Bronze Age agro-pastoral communities in southeastern Arabia that experienced the 4.2 BP RCC, or rapid climate change event, as well. Biogeochemical evidence from individuals interred at the Shimal necropolis during the Nar period from 2700 to 2000 BC and the Wadi Souk period from 2000 to 1600 BC indicate that this population did not experience a radical shift in diet or in infectious disease risk or other measures of human health during this time, despite increasing aridification. Instead, the inhabitants of this region appear to have responded to regional climatic pressures and economic shifts through population dispersal and decentralization into smaller, more stable communities. This was likely made possible because of relatively less rigid and hierarchical social structures than what we see in the large-scale trading partner in the Indus Valley. However, in our most recent work, my colleague Kim Williams and I have been working at the site of Dahwa in Oman. And the people of Dahwa adopted a very different strategy with very different results. Dahwa was a copper trading outpost for the Indus civilization from 4200 BP to about 4000 BP. They dealt with climatic uncertainty through diversifying their economic system. We have evidence for Indus ceramics, iconography, Indus script among the artifacts from the tomb at Dahwa, and we believe that this was a major center for redistribution of copper to Indus cities. This increased trade may have had dire health consequences. In a recent analysis of the human remains from the tomb and a bone pit that's associated, we've discovered that increasing reliance on trade in this one community in Arabia fostered the migration of mycobacterial pathogens. Despite the small size of this community with roughly 200 people in the tomb and associated bone pit, 40 of them have signs of maxillary infection, 26 have signs of severe infection, and 12 have signs that are highly pathognomic for leprosy. We are, of course, working to understand more about this site. Currently, my PhD student, Sangeeta Mahajan, is doing isotopic analysis using strontium, oxygen, carbon, and nitrogen isotopes. She compiled published data from Northwest India, Gujarat, and Oman, and she has found that these regions experienced 3 distinct groups in terms of their strontium isotope values. When she looked at the site of Dahwa, this is her preliminary data for her dissertation, so this is from 38 individuals at Dahwa and 10 individuals from a nearby site of Bat. She found that the people of Dahwa were comparatively a sedentary population in terms of their oxygen isotopes, but with various places of original origin according to the strontium isotopes, which was quite different from the site of Bat, where we had a highly local, mobile population, but they were of local origin. We're also using dental calculus from the site of Dahua to understand more about occupation and diet at this site. We know that a handful of people who were buried here were immigrants from Indus civilization, and so we are trying to characterize the mobility at the site using the isotopic ratios, characterize diet using carbon and nitrogen ratios, which we don't have preliminary data for that yet, I'm sorry. And we are also sequencing ancient DNA that was present in the calculus. That work is being done at Penn State University. So we have 9 individuals who had dental calculus that we examined in the scanning electron microscope using EDS and EDX. My graduate student, Darius Thompson, did this work. He found evidence for copper smelting, bronze smelting, as well as trade in gold and silver. He found thread from jute, silk, and cotton, and we found food refuse from Indian millet, okra, and turmeric. So we will be analyzing additional teeth to get a more complete picture of diet and the artifacts that are in the mineralized plaque deposits. And as I mentioned, we did extractions. This is my colleague Laura Wyrick and my lab tech Charlotte Shore. We conducted extractions for ancient DNA from the calculus of 48 teeth from the site of Dahua. The results for library prep and whole genome sequencing are very much awaited from Laura. And we will also be doing isotopic analysis on those same 48 teeth to look at the DNA from any pathogens, as well as the oral microbiome and food refuse from these 48 individuals as well. So paleopathology also has hopeful stories about small-scale communities who retained traditional knowledge and were able to diversify their food system in ways that created a lot of resilience. So in China, for example, paleoclimatological proxy data suggests fluctuating temperatures and changing patterns of precipitation and moisture in the Heshi corridor during the same 4.2 Ka BP event. As the environment became more arid, human skeletal remains from this region demonstrate no evidence of somatic growth disruption or pathophysiological changes like those that were discussed earlier today. Stable isotope analysis of human skeletons from this site has demonstrated that resilience meant selectively incorporating new plants and animals in the diet as a response to changing conditions. And adaptive diversity and intergenerational maintenance of traditional knowledge of local food options successfully buffered this population from nutritional insufficiency. So archaeology demonstrates a lot of nuance in the long-term health outcomes of climate change as they're mediated by environmental, biocultural, and social factors. While civilizational collapse and widespread environmental migration can happen, peaceful social and economic reorganization is also quite possible in the face of crisis. It's happened before, particularly in smaller, more flexible populations who retain traditional knowledge of ecological systems and the capacity for adaptive diversity and flexibility. I'd like to call for, as you all are conducting, interdisciplinary research to understand these relationships. We need to integrate archaeological, paleo-environmental, and paleopathological evidence. These diverse data sets are the way to address contemporary public health and policy concerns. And this work should be done through interdisciplinary collaboration. We have to draw on expertise from archaeology, environmental science, anthropology, epidemiology, evolutionary and molecular biology, and public health. and embrace a One Health concept, which is the interconnectedness of human, animal, and environmental health, which aligns with a One Health approach. Planetary health is something that I've become increasingly interested in as well, which adds political economy and an explicit critique of capitalism and other kinds of short-term thinking. One paleopathology is the way that we can build an ethical paleopathology. One Health has emerged as a non-anthropocentric framework to bridge the natural and cultural world and break down professional silos, but it is myopic without indigenous local knowledge and the concept of the Anthropocene to build a deep time perspective on human variation. And One Paleopathology and planetary health are thus the key to creating ethical, equitable, just, and sustainable futures for life on Earth. And while billionaires in my country contemplate life on the moon or on Mars, other countries such as China are rapidly adopting renewable energy sources to meet their climate targets. They're transitioning away from fossil fuels to proactively reimagine human life on Earth, starting with the principle that humans are biocultural organisms deeply connected to other species and to Earth's ecosystem. And I just want to say thank you to all of my collaborators, funding organizations, my graduate students, and my lab tech. Thank you so much.
Thank you so much, Gwen, for that fantastic lecture. We will move on to the next series of talks, and then at the very end, we'll come back and have a full discussion, because I'm sure that everyone has a lot of questions for you. So I think, Marco, are you presenting 2 in a row now? Okay, perfect. Okay, so I'll introduce, this is Dr. Marco Malela, I hope I said it correct, I apologize, from the Department of Biology at the University of Pisa. And we'll just quickly set up his presentation, so it'll just be a moment.
So thanks first to the organizers and for having us here in this awesome hall. And as mentioned, I'm a senior researcher at the University of Pisa since 3 months about. Before that, I spent 15 years in Switzerland, first in Zurich, then in Bern. We will touch some of these geographic settings during this talk. The first talk actually was meant to be presented by our colleagues from Granada, Jose Francisco and Silvia, which unfortunately cannot be here, so you're stuck with me for 2 talks in a row. Okay, the first one, but actually it's interesting because the 2 talks, they touch similar topics by applying on different geographical contexts and by applying a kind of zoom in, zoom out analytical approach. So the first one, which is a research that we published last year, and is focused on the medieval times in Spain, Iberian Peninsula, for a time frame encompassing from the 8th to the 15th century CE. And why is that? Well, because it's a chronological and geographical context which has several interesting features, while it includes at least a couple, if not 3, main climatic fluctuations. And also from a cultural point of view, especially when you compare the north of the peninsula with the south, you have a really interesting, intriguing cultural structuring. So in the north, we have Christian communities, where in the south, as you will see, Muslim regions. And of course, we have an even more contextualized heterogeneity, social heterogeneity, economic heterogeneity, which includes both rural and urban contexts. All this to basically test a main research question, which is actually what we are all interested interested about here. So which kind of interface between culture and climate and environmental features, especially climate, can we detect if focusing on stable isotope data concerning diet, mobility, and lifestyle for this specific geographic and chronological context. So in order to explore this main theme, we are addressing two research questions. We are testing two hypotheses. The first one more based on the type of influences that cultural and environmental variability have play on the north-south possible gradient, if there is a gradient in carbon isotope data. And the second one hypothesis is that given this climatic, non-climatic fluctuation through time, we expect to detect also similar or at least correlated trends isotopically. So the two, as you see already, the two variables, isotopic variables that we are interested about, that we are using as proxy of diet and the influence of the environment are carbon and oxygen isotopic, stable isotope ratio. Context-wise, this study considers 8 contexts, 4 and 4 for the north and the south. And you see that here we have, of course, as mentioned before, interesting differences, not only chronological, you will see better that in the next slide, but also cultural. So in the north, we have mostly Christian communities, whereas in the south we are dealing with Muslim ones. So which kind of influence the intersection, and which kind of intersection can we detect between environment, culture, and what we observe isotopically? So these slides presents, we are focused on adults only, and these slides presents the ratio between males and females in each context presented before. Whereas this one, it's a kind of sketch regarding the relative chronology of the use of each cemetery. So you see that there is for sure some overlap. However, the southern ones tends to be used for a relatively later chronological frame compared with the northern one. And this is interesting because we are going to discuss the results, also the possible link of the results with this kind of chronological sequence, or pseudo-sequence if you want. This was supposed to be the first speaker today, the first speaker of this talk, by the way. And the two variables that we are going to discuss are carbon and oxygen stable isotopes from animal. In terms of comparative data, we used both modeled and published data for the area investigated. We don't have faunal dental material to compare with. Okay, let's jump to the results. Here you have the geographical distribution of the 8 sites, and the heat map shows you how the average carbon value geographically. So the first thing that you notice already is that we have a pretty much nicely structured north-south gradient with relatively less negative values for the south and definitely more negative in the north. This is not only a geographic difference, by the way, but also an economic difference, if you want. Even I'm anticipating discussion parts, but this is the same plot but subdividing the sample between the two types regionally, if you want, but also including a second variable that is the difference between rural and urban context. And you see that when you focus, in both sexes, when you focus on the southern sides, the rural one are indeed the one with the less negative isotopic values, with the strongest signal of more important dietary portion of C4 plants. Now, of course, your question will be, what about difference between sexes? Well, difference between sexes are almost absent, both for carbon and for oxygen. You can see also here, and this is somehow the same results in a different way. Only San Badelio in the north shows a statistically significant contrast between the two sexes, indicative of differential access to dietary sources. These are the results for oxygen. Okay, so here the results are definitely less straightforward. We would have expected, basically, based on known climatic transitions, a chronological pattern, right? So basically reflected by more negative results in the southern and less negative in the north. But actually, what we observe, it's a mess. It's a mess. I cannot describe it otherwise. So basically, we don't detect any chronological pattern, but what we observe is the influence of local topographical features. So you see that there is okay, it's not super correlated, but there is a clear correlation between oxygen values and the altitude of the locality, which is not really ground shaking, if you want. But this just to say that this is something that militates against any kind of simplistic interpretation of the isotopic data in terms of a big pattern, which actually militates against my next talk, but I mean, what can I say? So, same kind of comparison in oxygen, this time between the two sexes, as mentioned. We don't detect any kind of statistically significant difference. This plot is also useful because we can explore, it was not a major focus of the study, however, it's interesting nonetheless, since you pay the presence of outlier, possibly indicative of non-local individuals for the study area. And as you can see, we don't have so much, actually. We have almost only one or two. And so in terms of residential mobility in this community, it seems like they were relatively stable. What is interesting, however, is that when we observe, and these data are kind of hidden here, but the individuals definitely more to the threshold of the distribution, or the outliers are mostly females, which is quite intriguing. However, just going back to the first question, the main question is, can we actually assess the interface between climate, geography, culture, on diet and mobility? And the answer is somehow. We are basically trying to address one research question, and we end up, as usual, which is the cool thing of these kind of studies, with 5 more. So different variables, different effects emerge as relevant, interesting, and worth of further investigation. So the first hypothesis was of a link between both cultural, environmental features and variability, geographical variability in carbon values. And this actually was probably the most clear results, in the sense that, as you remember, we have this clear gradient which is probably quite related to the cultural differences. We know that the Arabs brought important C4 crops when in the peninsula, sorghum is one clear example. So are probably looking at this kind of cultural effects on the diet of the people of the peninsula. But of course, we also have to deal with important environmental, both on a broader and localized scale. And this is even more clear when we are dealing with oxygen, in the sense that, yes, it's true, we were expecting some more clear fluctuation in the isotopic values given known and supposed climatic fluctuation, but actually the local features and the sample size of the analyzed context, representing the analyzed context, complicates the matter, definitely. Plus, all the variables that I didn't speak about, which is all the various possibly confounding variables, breastfeeding, the type of water sources, of course. It's not that we are dealing with really, as usual, with perfect data sets. I mean, we have a lot of background noise. And of course, the fact that cemeteries are used for an extended time frame. So even in terms of changes, climatic changes, we have a kind of accumulation of effects through time in the end result we are going to analyze. So some acknowledgements about this research and concerning both collaborators and performed analysis. And if you're interested about the paper, this is a link to the PDF. Thank you.
Thank you, Marco. And we'll now prepare for your second presentation, and we'll do questions at the end.
Okay, so still me, and this time we jump to a different context from a chronological point of view. This is a really ambitious research project, hopefully not too naive and over-optimistic, that we are working on with the colleagues in Bern. So, the question is, something has been said indirectly or directly, explicitly or implicitly, several times over the day. Stable isotopes, isotopic analysis in bioarchaeology are used for a whole range of research, to address all kind of research questions concerning diet, concerning sex to food sources, the influence of social structures, social differentiation, to reconstruct times of breastfeeding, weaning, and of course mobility and so on. However, if you squeeze all these topics, the main topic is the interaction between humans and environment, right? And their environment, which we all know includes both the biological and the cultural one. What we study with isotopic variables are proxies of the intersection, of the interface between human and their environment, which we can reduce to three main components, which is the climate, culture, and the specifics, the specifics of where people lived. Okay, so when we started this project, you will see we had a bunch of isotopic data from human samples, and which covers a admittedly large chronological transect. And one of the questions, besides many other, was the possible relationship between what we were observing and changes in climate. So real extended data set. You will see covers from Neolithic up to modern times. And the question was, how do they relate to paleoclimatic models? So it's interesting because explicit comparison between isotopic and from human data, from human samples, and paleoclimatic models are not so often made. So a lot of times there are indirect comparison and otherwise more contextual in terms of location or time frame. So very few big-scale syntheses of this type of data are available so far. Of course, a lot of problems in this kind of approach. We all know the main risk is to be oversimplistic, right? To reduce interesting research question to stupid answer. But in any case, it has also some very interesting aspects. So what we wanted to try to do with this study was actually to explicitly compare the stable isotope data from human bone samples through time for a specific location with long-term shifts, long-term fluctuations for that location. And this was possible because in Bern we have a huge data set, isotopic data set, which has been accumulated over the last 10 years, which covers different time frames with, as you see here, different sample size by time frame, and mostly related to the central part of the Swiss area, the so-called Swiss plateau. Now, most of the human and faunal data points that I will speak about comes from this data set we have in Bern, which we complemented with published data, especially for the for the western part of the plateau. And this data set has the advantage of being archeologically well contextualized, anthropologically also. So we have some kind of control on possible confounding variables. Together with human, we also have faunal data sets for each time, even though you see the data set, the sample size are definitely smaller. So this is what we try to investigate, explore from a diachronic point of view, with a focus in this context on the climatic variables, possibly playing a role here. And I want to say in advance that I know that it sounds sometimes oversimplistic, but it's not. In the sense that in parallel to this, to the climatic focus, which I'm presenting here, we are also exploring more contextual and social variables possibly influencing what we see. In any case, here you have the sample size, which with important variability through time, but this is what we have. For the Swiss territory, Roman times are notoriously represented by small inhumation cemeteries, and early medieval ones also not really numerous. So the results I'm presenting here are basically based on the carbon and nitrogen stable isotope ratio from human and fauna, from Swiss context dating from the late Neolithic up to the 19th century, mostly from long bones or temporal bones, and also I will show you how they behave, if you want, how they trend when compared with paleoclimatic models for the same area, which I tried to reorder in terms of comparable chronologies. Okay, from a paleoclimatic point of view, here we have the Swiss area, which I plotted here in terms of temperature and rainfall. This is the Chelsea data set, which is basically a paleoclimatic model based on the trace 21K, but downscaled. So we are seeing actually the data set itself, the model is represented by 100 years and 1 square kilometer resolution, which is really cool if you're interested, especially in the middle late Holocene, or in a relatively narrow time scale. So we see relatively expected behavior of the models for the glacial maximum up to the late Holocene in terms of temperature warming up and also rainfall data averages. And here we have basically the same type of results in terms of, again, temperature and rainfall, but by archeological time frame, if you want. Here I defined a series of time frames in order to be comparable with the isotopic data. And this is an average of the model for different cities, for different stations, if you want, from different location of the Swiss plateau. So it's not only one location, latitude, longitude, but the average of different models for different, and you see well-known diachronic changes and fluctuation in terms of both temperature, something that we heard a lot today, and also in terms of rainfall, precipitation. Now, these are the carbon diachronic trends. In blue, the humans, and in gray, the fauna. The fauna includes mostly cattle and pigs. I know the problematics of this, but let's focus first on the humans data set. I remember these are only, I didn't mention actually, how I selected the humans. These are only individuals older than 3 years old, which I select in order to try to get rid of the possible background noise of breastfeeding especially on nitrogen data. And okay, so what is really cool, I think, is this curve that you observe through time with carbon. So starting from the Bronze Age, something that Alessandra Varalli already observed, but also following then with the late Iron Age and a bit between the Roman times, which I have to remember you, however, is represented by a tiny sample size. We have this nice curve describing basically less negative carbon isotopic ratios for then a kind of jump for the early medieval times to more negative and a relatively stable then trajectory after that. This is even cooler if you compare with the averages for the fauna. So this deviation, which I show even better here, these are the difference between fauna and human across contexts. And you see this trajectory that you see here in a way bivariate. Here you see only combining the two. So there is a kind of through time deflection. So there is, it's always fascinating comparing this type of data, because when you see this kind of deviation among humans, you notice that something is going on, right? So we will see later what that may mean. Okay. These are the same results for carbon. In blue, the fauna, and in pseudo red, the humans. And I plotted on the top and bottom the averages, again, from the paleoclimatic model for temperature and rainfall for the plateau. I repeat, these are averages for different locations. That was just in order to see how human isotopic data plot together with the trajectory through times, trying to compare with temperature and precipitation. Okay, these are the second part is about the nitrogen, and you see that here the diachronic trends are really different in the sense that we don't have this sharp deviation in the diachronic trajectory of humans compared with fauna. The biggest offset is observed for late Neolithic times, and but it's interesting in any case, something that we observed also for carbon, this sharp change happening between Roman late antiquity and early medieval times, which is summarized by this curve, which is represents basically the difference between the two trends. Okay, this I want to stress that these are is something that we are working on, both concerning the paleoclimatic model, but also the human isotopic data and the fauna isotopic data. What I want to do now is to include more variables and see how they may interact. However, these preliminary results actually point to a relative consistency between human isotopic data and non-climatic fluctuations, because we have, you remember these changes in the carbon may well be related both to the introduction of, or better to say, to an expansion in the farming strategy of the population with the gradual introduction of C4 plants in the diet and crop variability. However, with climatic changes, more open landscapes, higher productivity, and this also may be reflected in the nitrogen values in the sense of higher access to food, to animal proteins, as well as this drop that we observed corresponding between basically late antiquity, early Middle Ages, with where we observe a drop both in carbon and nitrogen values. Okay, this is the optimistic slides. This is the more realistic slide, if you want. The sample size is unequal. It will be better to have tons and tons of data points by periods, but this is what the archaeology in Switzerland can give us. Some time frames are famously underrepresented by inhumation in the Swiss territory, especially Roman times and Bronze Age and early Middle Age. What about socioeconomic differences in the same population, especially when we are going to observe differences in diets? Which kind of variability they introduce? But on the other hand, would that be so relevant when we apply such a broad scale perspective? And of course, it will be really alarming to interpret everything we observe from humans as a kind of a result to climatic features or environmental features. So the risk is always to fall in this kind of structuralism, if you want, that we have to be really aware. And so this is what I was referring before with the need for more complex and nuanced approaches, which makes this kind of research endeavor more difficult, of course, because many times we don't have the information we would like to have, but at the same time, I believe, really intriguing. Thank you.
Thank you so much, and thank you for giving 2 talks, Marco. You've done a lot of work today. So I'd like to invite anyone from the audience who has questions. We'll also begin our final discussion session, but if you have any particular questions to start.
Thank you, Marco, for both talks. I have a question for your second presentation about the big trends of the isotope data. And did you try to split the human data between the adults and non-adults to see the same pattern?
Not yet, but it's a nice idea. No, not yet, not yet.
Okay.
I did by sex.
Mm-hmm.
I didn't notice any difference from that point of view. And by adults against subadults, that will be interesting. This data set includes also only subadults older than 3 years old. Huh?
Yes, I saw from the slide, but I was wondering, even considering the type of sample, because from the slide, maybe I'm wrong, it says the long bone. So considering the non-adults and the long bone for the turnover, so I was wondering maybe just giving a try and
definitely, yeah, definitely. I mean, I can tell you what will happen is that since it's a relatively large sample size, but actually really small by statistical point of view, we will end up with any kind of trajectory will be kind of open to, but it's definitely something that I'm planning to do.
Okay, thank you.
Any other questions? Anyone in the audience have more questions? Also for Gwen at all? For you? Okay, perfect.
Hi, about your first presentation, the southern of Granada, the chronology was until the 17th century.
So basically the one, they are definitely the southern ones are later, but I don't remember the chronology of the. It's, I think, one of the first slides. That's
okay. That was the question when it was abandoned. Those burial sites.
Yeah. So these are based also on this. But they are. This is basically represents the use of the cemetery. In terms of the community, we don't know so much, but they represent more or less the end of the use of cemeteries, each end.
Okay, any other questions while we're here? No? Okay, I think that we can move on to the final discussion section. As we had, just for anyone online especially, our final speaker is ill, unfortunately, and can't present on the archaeobotanical remains. So we can jump ahead to the final discussion session, which is going to be led by Professor Chavarria when she's ready. But if anyone would like to have a quick break as well before we start, we could have 2 minutes.
I mean, I'm quite tired. I imagine you are also quite tired. And I couldn't stop making questions during our meeting, so I don't have much to say. Well, first of all, I think the seminar was quite good. We discussed a lot. We could compare and confrontate our data. Much data was, as I announced at the beginning, about the Biolalia or then the GEMS project. We saw how different methods and different results can be different, but at the same time compared. And we also, I think, on my opinion at least, the, let's say, invited talks were very appropriate on what we talk about. I just wanted to underline some problems or some perplexities that I still have, taking into account that I have to make the conclusions of the same project I'm in. So I'm going to do a bit the bad experience or the questions or things which I still have problems with, although I have even written about them, okay? So it's my own doubts. One of the things we said from the beginning is that we are talking about a climate change period, which is very short. This is something we said from the beginning. We discussed this with Valentina a lot. I told her I would like, or I wanted to work more on the later period, the LIA, the little ice age, because I know a lot about the Lalia, and so I wanted to do something new. But at the same time, she said, we have worked a lot on the LIA, we want to work on the other one. It's a very short period in reality, because we saw, Bianca showed a very nice graphic on this. It's a period which starts in 535, 45, and finishes in the middle of the 7th, maybe beginning of the 8th century. We are not sure about when the medieval warm period starts and how warm was it. I've read some new things on the last months in which it's not so clear how warm it was or if there were changes. So it's a bit complicated. It's also complicated because we have, and mostly the PISA team has seen that there are changes. There are some health issues, mostly linked with respiratory diseases and so on, but we have other problems in the same period. As Bianca also said, it's a period of huge political crisis. I don't want to be too woke. I think it's a political crisis. Economic problems and market changes also. No? We know that the main problem from the end of the Roman Empire on is the end of the Annona system, and so we know that C3 cereals start to be less distributed in the Mediterranean. So how many things can be linked to the Lalia and how many things to the more general crisis, we still don't know. And again, we see that this 6th century crisis starts, let's say, in the 2nd century and goes farther. I didn't know until quite some months, I would say, when I was preparing this, my medieval archaeology course these years, that from the 380, 390, there were already big crises in Italy, northern Italy, so the end of the empire starts quite earlier than maybe we thought. And the 8th century as beginning of the warm period maybe could be also, let's say, advanced. So it's a problem. It's difficult to date also our material. We saw it here. The Serbian colleagues showed also how difficult it is to say we are working with material of the 6th century. Oh, sometimes the 6th century, sometimes the 6th, 7. I think 6th, 7 is a good period because the 7th century is still climatic, unstable at least. But we don't have much C14 dates. I think this is a big problem that we should solve. At Padova, we are trying for some years to date each burial that we find. If we are excavating a cemetery, we try to date each burial, each one. It's expensive, okay, but each time less expensive. The problem is that the dating of C14 is not so clear. It's not like your burial is 640, between 607 or 750, who knows. So this is a big problem. On the diet, I asked about the C14 thing in Spain, the C4. It's a pity that we didn't have the contribution of Rotoli and Marinato, because they are working a lot on this. They proposed me to present it, as I have been working with them, but it's a very complicated subject. I mean, I know it has been examined for earlier periods, for Bronze Age and even earlier and so on. For the early Middle Ages, it's quite complicated. I mean, seeing differences between C3 and C4 has been also attributed to climatic change or to taxes influence, because somebody has said that C4 was not taxed. I have never found the written source which says this. Historians say, it's something that we use a lot. Historians say what historians and which what written source. I also heard from somebody, some researchers, that C4 plants, particularly millet, is more resistant to climatic adversity. Yeah, but not warm, not the dell'alia, climatic adversity. As I know, and maybe you can tell me for Spain, millet is more resistant to drought, to dry weather, to lack of water. And as we know, for the early Middle Ages, that was the contrary, which happened in northern Italy, for example. So why was more millet? Was it because of the climate change, or was it because other problems? I think it was other problems. I think there was less C3, for example, or it came with other populations. We know now that Lombards, for example, used to eat C3 also when they were in Pannonia. So I think the millet problem is something that we repeat and repeat and repeat again, and we should start seeing it from the beginning, maybe with the help of the dental calculus thing. You said you found millet. We'll see what kind of population eat millet. Do they all eat millet? Yeah, but if we find it in northern Italy, where does it come from? It's all the population that eat it. And the last problem I wanted to underline is the thing that all these kinds of analyses have been long developed and studied for prehistoric periods, maybe because it's more difficult to find written sources, and so to develop new methodologies at that time. We're trying to develop this for the Middle Ages, but we have a gap still. Not, it's being full, but we have still the gap in Roman time. So when we said we start, or there is a development of, we should be more careful and wait than classic archaeologists start publishing this kind of analysis to see if it's something new or it's simply something that haven't been studied yet. I think in general, we had many new ideas, many new things were published. As I said in the beginning, these are very preliminary results. We know also these PRIN projects last 2 years, which is very short time to develop this kind of research, knowing that, for example, DNA needs years from the moment that you sample until the time you have the results and interpretations, but it's not bad. I hope and I wish we have the opportunity to do another seminar still on this Biolalia project. As maybe some of you know, the project has been temporarily enlarged chronologically. We did this colloquium, this seminar now in May, because we did want to have papers for September and so on. We still want them, as I talk with many of you. I want your papers by latest September, because we want to have the journal. We will publish it in the Post-Classical Archaeologies journal in Italy. So for December must be out. We will do it. We will try to do referees between us or something. So, but possibly we could do another seminar in maybe January, in order that Valentina can also enjoy this and join. And who wants to add something? We didn't ask you things because generally nobody asked to the highest specialists because, and your paper was so clear. But what do you think about this seminar? Do you think something new was said? What would you, let's say, push to research? Are you going to live in Europe from now on? Pera told me she's not returning back with this paper.
Thank you. Sorry to disturb you.
Thank you so much. One thing that I think would be really interesting, if it's possible, is to incorporate analysis of the faunal remains. There were some analyses of faunal remains, even pathological analyses, and also ancient DNA, even looking at dental calculus from animals, I think is really an important area of research that's very neglected in paleopathology these days. And so I would really encourage everyone to think more about the animals, actually. I liked seeing that the isotopic analyses of animals had been done, but even, I don't think many people have looked at like cementum annulations for animals or looking at their dental calculus. I think it would be really interesting.
Thank you very much, Gwen. Does anyone else have any questions or comments, particularly for our keynote speaker? She came all the way from the USA. I actually have a comment for you, Emanuela. Basically, when you're doing your dental calculus research, have you also incorporated any form of like chemical analyses? So for example, I've worked a lot in the Eastern Mediterranean, so we have a lot of metalworking, metallurgy, and smelting and this kind of thing. And so we're always assuming that the individuals that engaged in these type of activities were also, of course, inhaling, ingesting, and absorbing these type of fumes as well, particularly when they're in like a city context. Have you been able to, or have you considered, I'm not sure about Italian context, I know that there's plenty, but I know access to material is a whole other thing. But have you thought about applying some of these techniques like XRF mapping or laser ablation or so on to kind of like chemically analyze some of the calculus in combination with the microscopy to see if you have like incorporation of metal elements as well or like toxic elements?
Yeah, this is a very, very interesting and good question. There are articles and specialists working on that, and I proposed like an example for the Bronze Age for lignite. So there has been, application of methodologies with gas chromatography, with pyrolysis, like gas chromatography. I didn't myself, probably mostly because I'm working with like earlier periods, so I didn't have this like necessity so far. But we are trying to apply this for the use of mercury, for the like as a medicine, as like a medicament, and has been, it's now under review with specific chemical analysis, like, and it seems like it works. The only problem here is like the reference collection and also contamination coming from the sediment. And this is a topic that's been kind of trying to understand how to take over this problem, maybe making some kind of experimental work on taphonomy diagenesis in sediment, like experimentally created to understand how, for example, metals or minerals in sediment can penetrate, because after all, the dental calculus is a porous and kind of it can get like in infiltration, and so to understand how this works is still like very open, and the problem of contamination is important. And this is a problem for archaeology, but it's also a problem for forensic analysis as well, because once we understand this, we understand also, for example, provenance. Because I just like make another question. If I think about like analyzing these in mineral-rich environment or sediments, like where it's already naturally rich in some sort of copper or something, how can you kind of distinguish what comes from the sediment and what comes from what you drink? Or it's very difficult. We try to overcome this, maybe suggest trying, and that's what we would like to do, to replicate some kind of modern and lab done calculus, like with precipitation of salts and saliva, and so create like a fake calculus, and then understanding different type of sediments, so kind of see how it behaves, and then analyzing how because this is also important not only for archaeology, but also if you're thinking like in medicine and processes and everything. So how, for example, you insert something in your jaw as a process, and how this like entails with your microbiome, and so it's a very important question, and I didn't do myself, like we are trying to do experimentally, but I haven't applied this just for mercury, and it's in more recent, actually it was not me, it was a mercury I had in the lab, Elena Fiorin, and she is now publishing the results for the 17th century people affected by leprosy, and it seems like some good results is coming with regards to mercury, but this is it.
Yeah, thank you very much. And it's like, I'm really glad actually you said that was your response, because we need these experimental studies, because something that I'm seeing increasingly in paleopathology, and my colleagues are going to be like rolling their eyes because they hear me complaining about it all the time, is the misuse of some certain techniques on bone and teeth. Like, for example, the only way we can tell endogenous versus exogenous incorporation of elements is to literally use like synchrotron level techniques to look at like the incorporation of elements into the osteon, even in bone. There is no way to differentiate with exogenous versus endogenous macroscopically, just doing things attached to the bone. So I'm really glad that you're actually doing that with dental calculus now, and you're looking into it. Have you thought about trying to connect? This is just something that came to my mind when you were speaking. Maybe with some of the body farms, like even the ones that they have now in Europe, from the forensic context, because maybe they have individuals that do have dental calculus in this controlled environment, that they're already doing the taphonomic studies. You might be able to find a nice collaboration there. Just a thought.
Actually, yeah, even this is a very good suggestion, because like this observation, and that was saying from a collaboration with forensic forensic odontoiatrists, and because this is important for both the field of studies. So if it works with the forensic and works with the engine, like it's, so yes.
Oh, thank you. Yes,
I'm just.
I really feel like dental calculus is this kind of tool that, like, although we've used it a bit in the past, I feel like it hasn't completely been unlocked yet. And I think I saw Gwen in your presentation. You had a lot of SEM analyses as well, and so that's quite interesting to see the application of the scientific techniques. Does anyone else have a?
I'll just mention one thing about the scanning electron microscopy, because in that case, you can actually see where the particles are embedded in the calculus, because we haven't dissolved it. And so if we're looking at a piece of tin oxide and it's buried deep inside the calculus, it's a different matter than if it's on the surface.
Oh, so this is a great way. It's kind of like stratigraphy of the dental calculus, but you need it like intact, like, because we're so used to sampling and like taking it off immediately, but you need it not touched in that case.
Yeah, so we actually look at it with the scanning electron microscope and use EDX, and then remove it and do the DNA.
Okay, great. So 2 birds, 1 stone effectively. Okay, thank you so much. Does anyone else have any questions or comments or discussion points that they want to raise from any of today's presentations or future research prospects? No? Okay.
I think another thing which we should and show as an example of this project is that different universities working exactly on the same thing, because Pisa and Padova are working on paleopatology, in both cases, archeology, in both cases, stable isotopes, can collaborate and not be, let's say, concurrence. And I think this has been also very good, working together, learning from both groups together, different techniques. Bianca has come to Padova, we have come to Pisa, and so on. I think this has, we should say it's Pisa, Padova, and Milan, because one of the members of the Padova team work for the laboratory also in Milan. I think this should be wider done in Italy on this subject, because we can study, I told somebody before, Roman villas. Everybody can study a Roman villa, but we cannot study all together one skeleton, because it's one university which has it, and they will never let you study it. So working together in this subject, I think it can give us many new opportunities in future, and we showed that it's possible and enriching and everything. Well, I think we are.
Yeah, I think we're ready to conclude today's talks in that case. And for any of you that are still hanging around, even for tonight as well, we'll take you to the museum exhibition. I don't remember the name of the exhibition, I'm sorry, but it was curated by Valentina and the rest of the team in Pisa as well. Sorry, I wasn't involved in the creation of it. It's not too far. It's just past our lab at Via Derna. So basically, we'll take you there.
What time?
We're heading there basically straight after this. So if you'd like to come with us, we can all walk together. And then we can also stop by our laboratories in Via Derna and have a look at our anatomical collection. And then we'll see where the night takes the rest of us. Yes, with the social dinner and so on. All right, thank you everyone for attending, and thank you for your patience. So greatly appreciate it.